Good morning to everyone. I am going to take the monoclonal antibiotic. I am in Department of Pharmaceutical Biotechnology. That monoclonal antibiotic means a single clone of the antibiotic. That is antibiotic means it is the glycoprotein which is secreted by using that antigen. Antigen other name is the immunogen. Immunogen that can be converted into that immunoglobulin, that is the antibiotic. The immunoglobulin that can be classified into 5 types. One is the immunoglobulin G, immunoglobulin A, immunoglobulin M, immunoglobulin E, and immunoglobulin D. The monoclonal antibiotic that is invented by George and Caesar in 1975. The technique used in that is the hybridoma technology, which is used to produce that monoclonal antibody. In this case, immunogen we are introduced into the body, produce that immunoglobulin. That is followed by the fusing of the normal cell interact with the myeloma cells. Then fusing by using that polyethylene glycol. Before that we are using that as antivirus. Polyethylene glycol which is used to fuse that normal cell line, and deficiency of HGPRT cancerous cell line. HGPRT means that is the hypogenin guanine phosphoribosal transferase enzyme, which is mainly involved in that hybrid cells. So first step is the immunization. Immunization means the artificially active that substance, that is the vaccine, that is introduced into the mice. Then after that it produces that antibody. After that the spleen cells that can more amount of B lymphocytes which is involved in production of antibody. Then fuse with the B cells and myeloma cells. Then these hybrid cells that can be selected by using that selective media, that is the HAT media. HAT media means hypogenin, aminoterrin and thymidine media, which is used to separate only the HGBRT enzyme. So that can be analyzed by using that ELISA method, that is the enzyme linked immunosorbent assay method. The selection depends upon the synthesis of nucleotide by mammalian cells. The mammalian cells can synthesize nucleotide by two different pathways. One is the de novo pathway, second one is the salvage pathway. First one is the de novo pathway. De novo pathway, it needs that dihydrofolate reductase enzyme, which is involved to produce the tetrahydrofolate derivatives. This tetrahydrofolate that is present in 3 places. First 2 places, 2 of the carbon in purine ring, that is provided by N10 formyl tetrahydrofolate. Then methylene tetrahydrofolate, which is involved in conversion of the deoxyuridine monophosphate converted into the thymidine monophosphate. The folic acid analog, that is the aminoterrin, that is present in that selective media, which is involved to inhibit the production of the tetrahydrofolate, which is provided in the tetrahydrofolate enzyme. Next one is this aminoterrin, it is a folic acid analog, so it is block the de novo pathway. So automatically it reaches that salvage pathway. In this salvage pathway, 2 types of enzyme involved in that salvage pathway. One is the HGPRT, second one is the thymidine kinase. HGPRT that is involved to convert the guanine to guanine monophosphate and hypogenin to inosine monophosphate, and thymidine kinase, which is involved to conversion of the thymidine to thymidine monophosphate. Okay. Next, this can be this monoclonal antibody that can be directed by using that ELISA method. ELISA method, there are three types. One is the indirect ELISA method, second one is the sandwich ELISA method, third one is the competitive ELISA method. In that indirect ELISA method, the micro titer plate, 96 well micro titer plate that can be coated with the antigen. Then the specific antibody, the measured antibody that is transferred into the 96 well. After that, was the solution. Then after that, adding that enzyme linked conjugated secondary antibody. Then after that, we are using that substrate, that is a chromogen substance that is used to convert into the color change. Okay, that one is the indirect ELISA method. Second one is the sandwich ELISA method. Sandwich ELISA method means instead of that antigen, we are using that antibody coated well. Then again we add the measured antigen. After that, we add that enzyme conjugated secondary antibody. Then we add the substrate to color change. Then competitive ELISA, the antigen and the antibody both are incubated in a separate flask. Then we transfer into the 96 well already coated with antigen. Then we add that enzyme conjugated antibody, secondary antibody, then substrate. Here we are using that quartz peroxidase, alkaline phosphatase and beta galactosidase as a enzyme. Enzyme linked immunosorbent assay, that is relevant to that RIA method, that is the radioimmunoassay method. Radioimmunoassay method, we are using that radioisotope. Here we are using the enzyme instead of that radioisotopes. Okay.